So, what if you could edit the very code of life itself? I'm not talking science fiction. A technology exists right now that gives us that power. It literally puts evolution directly into our hands. It's called CRISPR, and believe me, it's already changing our world in ways that are just absolutely profound. All right, so here's our plan. We're going to start with the problem that CRISPR solves, why we needed it. Then we'll dig into the tool itself. After that, we'll see its life-changing impact, look at what's possible beyond just medicine. And finally, we have to face the huge, profound questions this all brings up. Let's dive in. So, first up, reading versus writing DNA. You see, for the longest time, our own genetic code was basically a locked book that we could only read. And just imagine how frustrating that was. For decades, scientists could see the four-letter code that dictates everything about us, from our eye color to our risk for Alzheimer's. They could even pinpoint the exact typo causing a horrible disease. But the power to go in and actually fix it, that was pure science fiction. So this blueprint for life was essentially locked down. Now, there were a few early gene editing tools, sure, but they were like trying to perform surgery with a hammer and chisel. Just clunky, really inefficient, and incredibly difficult to use. And this is where you really see the massive leap forward. The old tools were bespoke, meaning you had to build a unique, super expensive tool for every single tiny change you wanted to make. CRISPR, though, it's programmable, it's quick, and it's precise. It's like we went from carving letters in stone to suddenly having a find and replace function for the code of life itself. A total game changer. Which brings us to section 2, CRISPR, the genetic editor. So how does this revolutionary tool actually work? How does it rewrite life's code? Well, it's actually this beautiful, elegant, two-part system that scientists figured out by watching how bacteria fight off viruses. First, you have a guide molecule that's programmed to find one exact spot in our 3 billion-letter genome. Think about that precision. Then a protein swoops in like a pair of molecular scissors and makes a clean cut. The cell's own repair system then rushes in to fix the break, and boom, that's our window of opportunity to edit the code. And as Nobel laureate Jennifer Doudna puts it, that programmability is everything. It's the key that unlocks the entire genome. It's what makes CRISPR not just another discovery, but a true technological revolution. And this isn't some far-off, futuristic idea. Let's talk about how CRISPR is already changing lives, right now. We'll start with a disease that, until very recently, was considered a painful life sentence. This is Victoria Gray. She lived with sickle cell disease, a terrible condition caused by a single-letter typo in her genetic code. Just one tiny error, and it led to a lifetime of debilitating pain and constant medical treatments. And the genius of this therapy is how they did it. It's a one-and-done treatment. Instead of trying to fix the faulty gene, scientists used CRISPR to simply turn on a different, helpful gene that we all have. This gene makes something called fetal hemoglobin, which basically cancels out the effects of the disease. And the result for Victoria? She hasn't had a single pain crisis since the treatment. It's just incredible. The outcome, as you can imagine, is just profound. For Victoria and the other people in this trial, this isn't just a treatment, it is, quite literally, a second chance at life itself. But the potential here goes way beyond curing genetic diseases in a few people. CRISPR is really poised to reshape our entire world. I mean, the potential applications are just staggering. They pretty much fall into 3 big arenas: completely transforming human health, revolutionizing agriculture, and even helping us tackle some huge environmental challenges. So in agriculture, for instance, we can make precise changes to crops that used to take decades. For the climate, scientists are editing the microbes in a cow's stomach to cut down on methane emissions. And get this, we could even edit the microbes living inside us to prevent diseases like asthma without ever touching our own human DNA. The possibilities are wild. But with this incredible new power, well, it forces us to confront some of the biggest questions humanity has ever faced. The question is no longer if we can change our genes. The question now is how, and if we should. Okay, so the most crucial distinction we absolutely have to understand is this: somatic editing versus germline editing. Somatic editing, like what Victoria Gray had, it only affects the cells of one person. The change ends with them. But germline editing, that alters reproductive cells. That means the change is permanent. It gets passed down to all future generations, forever altering the human gene pool. But, for people who are actually suffering from these hereditary diseases, these big ethical debates can feel a bit abstract. As Huntington's patient advocate Charles Sabine points out, when you're facing the reality of a devastating illness, the moral choice to act can seem crystal clear. And this is a real challenge. Where do we draw the line between a treatment and an enhancement? Let's say we use CRISPR to make sure someone never develops high cholesterol. Is that treating a future disease, or is it enhancing an otherwise healthy person? That line gets very blurry, very, very fast. And all of this leads to maybe the most important question of all. Who gets to decide? Who makes these choices for all of humanity? Who's responsible for charting the course of our own evolution from here on out? This technology places our genetic destiny squarely in our own hands. And as Jennifer Doudna writes, that power is both awesome and terrifying. Deciding how we handle it may just be the single biggest challenge we have ever faced as a species, and it's one we are all going to have to face together.